I'm Mariano Soiza Reilly. I'm from Argentina, from the beautiful Buenos Aires. You have here a picture of my institute in a sunny day as today. So many days during the year are like this. This is not fake or filters or anything weird, you know, it's like this. So in our lab is located in this institute called Instituto de Fisiología, Biología Molecular and Neurosciences, Neurociencias. And so we are interested in the lab in applying especially array tomography to try to understand corticolimbic circuits, specific circuits that are linked to emotional control and mood disorders, involving mood disorders. So we are interested in that. And what I'm going to talk today is how we started.
Mariano, can I stop you? You are
Yes.
showing the. You are not showing the full screen right now.
Sorry.
Yeah.
Sorry.
So we can. I think we are seeing the presenter mail.
Yeah, wait, because I have this. Yeah, I have my second monitor. Sorry. Yeah, sorry.
Good.
Like this is fine.
Yeah.
No?
Good.
Okay. Thank you. So I'm going to show you a little bit how we use this. I was saying this technique, array tomography, to study input-specific brain circuits. Can you hear me? Here is frozen. Can you hear me?
We hear you well.
Okay.
Yeah.
Sorry.
Yeah.
So then, okay, I'm gonna go through this. Otherwise, I'm gonna be late. So everything started with the seminal paper of Christina Micheva and Stephen Smith in 2007, where I knew for the first time about array tomography in this neuron paper. It's a beautiful paper. And from there, at that time, a little bit after actually, I was in contact with Christina, with Stephen at MBL in Woods Hole. I was a student in a course and I had Christina as a professor there, and she showed me actually for the first time array tomography as a student. I was kind of in love with this approach. Later on during postdoc, I started using this technique. So here is a bit of summary of the main steps that we have in array tomography. This is a high resolution immunofluorescence technique. Then others are going to talk about the potential for correlative with EM. But the basic idea is we start with the fixed tissue embedded in a plastic resin. In my case, I use LR white. And then after curation, we can go for ultra-thin serial sectioning, typically 70 nanometers. We could go up to 50. And then for this serial section, we use diamond knife, of course, but this particular is the jumbo knife that allows to collect very long serial sections. I mean, ribbons of serial sections. Then these serial sections are transferred to a cover slip. And once the sections are on the cover slip, they are pretty stable for conventional immunolabeling. Then we go for acquisition of serial images. And then with the computer, and most of the things we use is with the ImageJ through using Fiji and different plugins. We basically align these serial sections and reconstruct the 3D view of these stainings, okay? This is a general overview of the technique. Okay, this is a little video that you probably are familiar with, and of showing the serial sectioning. And then you see here, I don't know if you see the pointer, but you see clearly the sections coming through and one attached to the other. To be sure about that, what we do is in the block, we put glue on the top and glue below of the block face, and then subsequently the slices get attached to each other. And then we got very stable long ribbons, okay? Okay, I put this one just because I don't know about who is familiar with the immunofluorescence basics. This is a very, very basics, but the idea is that on any tissue or cells or whatever source biological tissue that you are managing, you have antigens, typically proteins, lipidic molecules, or sugars, and then we're gonna use a primary antibody that is going to recognize antigens, one of these antigens in the tissue of actually our antigen of interest. Then this primary at the same time can be recognized by a secondary antibody that is typically conjugated with a fluorophore, with a molecule that under particular excitation circumstances can give us a fluorescent color. And then exactly as I was saying, you illuminate these fluorophores and then you get the fluorescence. Okay, this is basics. So as I was, maybe I didn't mention, but array tomography has a great benefit of resolution. The resolution is very high, even in comparison to any confocal microscope. Here is a very nice example of Wong and Smith paper. Actually, they compare this resolution, especially in the z-axis, because in the xy axis resolution is pretty similar to AB's resolution, but when you go to z-axis, you see how we are far below the resolution of the AB's maximum resolution. This is because actually our resolution in the z-axis is given by the physical sectioning of the sample. So then as thin as you cut, this is the resolution you will get. So then you can go up to 50 or 30 or 40. Then, of course, the challenge is to detect the fluorescent signal, okay? Because the emission is very low. But typically, it can go between 10 and 15 times lower than the resolution you get with the confocal. This is a little bit of overview. Okay, so this is an example of what I'm working on. Actually, this is a serotonin neurons in the dorsal raphe of a rat, and then array, because of this resolution that you get, you can identify individual synaptic buttons. These synaptic buttons are identified by the protein synapsin. Synapsin is a structural protein in the synaptic vesicles. So then you can identify these buttons. And for instance, here you can see individual synaptic buttons on top of a piece of dendrite of a serotonin neuron. So then great advantage of AT is that you can do quantitative analysis in the volumetric space, I mean the whole tissue of these individually identified objects related. You can do the global analysis versus the ones that are only associated to, for instance, serotonin neurons. So then you can analyze somehow different populations of synaptic buttons. This is what we are doing in the lab. Okay, here's an example of volume. I don't know if it's going, if it's moving properly online. Here looks pretty slow, probably the connection. But the important thing about this, I'm showing this because I'm sure you are familiar with the volumetric analysis when you do confocal. And then as far as you get deeper and deeper in the tissue, you start seeing that the blur and the darkness is appearing. So with array tomography, the good thing is the resolution is given by the physical sectioning, and then on each section, the immune staining is, we say, is isotropic. So then the antibodies, they don't have a penetrancy issue. So then the let's say the quality of your stack is isotropic. You get very high quality at all the levels, even if you go deeper and deeper, even with, I don't know, thousands of sections, okay? Well, and one of the properties that we use a lot and actually makes array tomography very strong, I think, is the capacity of multiplexing, of multiple labeling. This means that you can run an immuno with, let's say, with your 3 antibodies, reveal that, take the picture, serial pictures, then you can elute off those antibodies and rerun a new staining with 3 new antibodies. So then at the end, you collect volumetric images containing all those stainings in the same tissue, in the same sample. It's not that you need to go to adjacent sections to get staining with a 4th marker, let's say. You can put all these markers together at the same time, and then you can study associations to each other in the same tissue volume. So this is a very strong capacity that you get with array tomography, and this is what we are really exploiting in the lab. Okay, but then how do we use this method to study synaptic circuits? We saw how we label maybe synaptic puncta, synaptic objects, but then how do we study synaptic circuits? Okay, so I put a classical EM image of a real synapse, how it looks, you know, with the presynaptic terminal and the postsynaptic piece of dendrite with the clear postsynaptic density region. So then, okay, we have the synapse here. So how do we do to study this? Because this synapse is very clear. However, we get a huge variety, a huge diversity of synaptic contacts in the brain, for instance. Okay, so then these are all different synapses. And if you see, they are more asymmetric. They have they are partitioned. They are like dual or triple. So how do we use array to study all of this? So one way to do this is, okay, how do we study diversity? So one way to do this is to actually to have tools to be able to identify different, at least neurochemically, different synapses that might have different properties. And this is the case of array tomography. Array tomography is taking advantage of this is a synaptic button. This paper is amazing, the inner life of the synapse. So what they did is a quantitative proteomic analysis of synaptic button. So then you have all, I mean, the numbers, you get the actual density in this synaptic button of all these protein content. So then with these components, you can identify some of these components that are general for all the synaptic buttons, and some of them are selective, let's say, for glutamatergic or for GABAergic terminals. So then, taking advantage of this proteomic knowledge on synapses is what we use in array tomography. So basically, here is an example where I studied the dorsal raphe in the mouse in this case, and the idea was to study how is the synaptic neuropil organized in the raphe. And to do that, what we did is to identify, because the dorsal raphe is a mess, it's like a bag receiving multiple sources of inputs, some of them glutamatergic, some of them GABAergic, and multiple neuromodulators. So then what we did is to, using these specific markers, for instance, the vesicular glutamate transporter 1 or 2 or 3, we can, or even GABA or GAD, the enzyme that is in charge of the GABA synthesis. What we did is to identify these multiple sources and study and make like a populational study of these inputs. So, and to do that, of course, we did it with array tomography. Here is an example of synaptic buttons that are, they have, all of them, they have synapsin. We usually quantify a synaptic button. We consider a synaptic button when they have synapsin, but also they have a specific marker for the neurotransmitter system. In this case, what we use is bglut1 or bglut2 or bglut3 for glutamatergic inputs and GAD2 for GABA inputs. So here you see across the section how the synaptic button appear, and okay, this is not disappearing, but some of them usually across serial sections you start seeing the button, and then you the button seems to disappear. This is because you are chopping across the synaptic button, okay? And the orientation is completely stochastic. So in this study, we quantified these different populations in the tissue volume, and what we found is that most of the inputs were BGLU2 positive or GABA positive, meaning that most of the inputs to the RAFE are coming from BGLU2 sources, BGLU2 containing cells as a source, or GABAergic sources. And from what we know of BGLU2 expression, BGLU2 is mostly expressed in subcortical regions, meaning that most of the glutamatergic inputs to the RAFE are subcortical, okay? While GABAergic, they are most of them subcortical as well. Okay, but then we went a little further with this, is because we started actually by looking at these stacks, we started looking at weird things. Weird things meaning that we saw a position very close to each other of glutamatergic butons with GABAergic butons, okay? And this happened for BGLU1, BGLU2, and BGLU3 butons. So then we quantify these instances of potential association between two butons. And what we found is that actually BGLU1, BGLU2, and BGLU3 butons were associated with GABA butons more often than between to each other, I would say. BGLU1, BGLU2, BGLU2. So then you had like a glutamate GABA association that was not really present between glutamate axons. So then we wanted to double check this, not because the only problem that you can have when you just count dots in the space is that just by chance you start seeing false positive associations. So what we did for this, to study if they were specific, we did a cross-correlation analysis of pixels. In this case, or in this analysis, maybe for some of you that are not very familiar with this analysis, what we take is two channels that we think they are associated, and we start moving apart one of the channels from the other. In this case, let's say we keep the blue channel fixed, and then we start physically moving away, giving a few microns of moving this channel. And then what you will expect to have is that if they are really tightly correlated, then this displacement, when you start displacing, this correlation should go down very, very deeply. So then we did this analysis for this GABA-glutamate association, but also for the glutamate-glutamate associations, and we found that here we have the values were more higher, the glutamate-GABA associations than the glutamate-glutamate. But because also, another step further, because also this could be sensitive of, this is absolute number of dots. So then if you have more dots, you have also more chance of having this, having fake association. So then what we did is to normalize with the, by the maximum value. And this is relative or normalized cross-correlation. And then like this, you see that the glutamate GABA associations have like a steepened, okay, curve in comparison to the glutamate association. So I like to talk about this because with this approach, actually with array tomography, we were able to find this apparently new morphological feature present in the RAFE. So then we wanted to look at it at the EM level, of course. So we did immuno-EM, and we found instances of this is a glutamatergic axon, this is immunolabeling against GAT2, and we found axons that were very close to each other, having a clear synaptic sites with on a postsynaptic dendrite, and then this maybe this picture is not the best, but there were some instances that looks like a symmetric synapse between these two axons. Then we wanted to go functional with this to see if this association could have any functional meaning. And then in collaboration with Chris Vaughan in Australia, what he did was patch clamp recording, ex vivo patch clamp recording, and what he found, to make a long story short, because this is about array tomography, what he found is actually when you manipulate the activity of the GABA axons, either through the GABA A receptor or GABA B receptor, you get enhancement or inhibition of glutamate release on the postsynaptic dendrite. So then GABA, through this axoaxonal association, could be modulating the glutamate release in the raphe. Okay, so this started 100% with array tomography. This is why I talk about this example. Okay, so now another great benefit of array tomography is we can study input-specific synaptic circuits, and this is kind of the title of the talk. And this is what we are doing most of the time in the lab. And for instance, in our case, we are interested in this particular circuit that is connecting the prefrontal cortex to the dorsal raphe nucleus in the mouse, because this circuit has to be is related to mood disorders. Okay, developmental changes in this circuit, we are studying that actually we have evidence that is linked to emotional alterations in the adulthood. Okay, so how do we study this? So here we did a little trick on the classical array tomography protocol of Christina and Stephen Smith. And so to preserve, in this case, we need to preserve endogenous fluorescence. I'm gonna show you why. Because we labeled this pathway before, but we need to preserve this fluorescence. We have to go up to 70% dehydration, 70% ethanol. We don't go to 100% ethanol because otherwise it's killing the fluorescence. And then the other little trick that we started using and doing is to do Eller-White flat embedding. Very similarly to what we do for EM or for immuno-EM, is to do flat embedding but with Eller-White embedded sections. And okay, here there is a book with a chapter explaining this in detail, but to go through pretty fast, what we do is to basically do the embedding in Eller-White as usual of a vibratome section, like for flat embedding. Then the tissue we put it on a petri dish, it's inverted petri dish, and then we use a cover slip or something to put some pressure just to be sure that the embedding is going to happen. I mean, the filtration especially is going to happen on a flat surface with some flat pressure on it. Okay, could be a cover slip with something on top of it. And then what we do is to prepare a little cassette, like a spacer of Aclar on top of a glass slide, spacer of Aclar with a little hole. In the hole, we put the Eller-White, we put the tissue, then we cover with Aclar again, and then again we put some pressure, and then we cure that. Okay, we put that in the oven. Then the sample is polymerizing really nicely. Here is an example of a sample, for instance, and you get very nice orientation of the circuit that you are interested on. Okay, either the target or the origin of the circuit. That when you do randomly in a capsule is really hard to be orientated afterwards when you have to section. So then this, what we do is to excise the little square to make this is gonna be, I mean, I have to clean this but afterwards, but this is gonna be the block face. And then in this case, you can see actually a dentate gyrus there, so you can very easily go to any specific layer there to analyze. So this block, I just glue it on with crazy glue on a top of a block of a harder resin, and then that is my sample to do the ultra thin serial sectioning. I'm going to show you a couple of examples. In these examples, what we do is to work on a SERT-Cree animal. It doesn't matter SERT what it is. It's a serotonin transporter, but the idea is this Cree animal allows me to inject a virus actually in the prefrontal cortex, and this upon this Cree recombination, this virus is going to start expressing TD tomato in those Cree cells. Because I know these Cree cells are going to the dorsal raphe, then what we can do is to analyze which for instance, which proportion of all the prefrontal cortical inputs that are revealed by BGLUT1, they are very specific for cortical inputs, are exclusively or selectively coming from SERT glutamate neurons. And so with this approach, as I mentioned before, of course, we need to preserve the fluorescence, the endogenous fluorescence. This is why we go up to 70, okay? But then we need to be sure that we are in the RAFE, we are in the right region in the RAFE, okay? And for that, we do the flat embedding. What we do is then with the tomato here, we see an example across serial sections, labeling the axon with the tomato, and then this axon at some levels, they have also synapsin, because these are supposed to be synaptic release regions. And then if you see, there is a co-localization with BGLUT1, because we expect that these cortical inputs, they express BGLUT1 at the synaptic release region. So you can follow this axonal button across the serial sections. So then we quantify these, and what we found is that, let's say, out of these BGLUT1 synapsin positive buttons, only 33% of these buttons were negative for T tomato, while 67% they have, of course, these two markers because they are coming from the prefrontal, but also they are T tomato because they are SERT-CRE. They express CRE. Okay, so then we can like this, we can do populational analysis of different synaptic buttons. Okay, here is, I have two examples, very short examples, but just to give you a sense of what we do, is we collaborate with people also interested in circuits, in input-specific modifications, with drug abuse, with mood disorders, and different kind of analysis. But for instance, with Frank, in this study, and Manuel Mameli, what we did is to they were trying to analyze the maladaptive changes in this particular synapse between entopeduncular nucleus and the lateral avenula during cocaine exposure. So then what we did is we found that these particular synapse that get sensitized or modified after cocaine, decrease actually after cocaine exposure, is a very particular synapse that co-release glutamate and GABA. And what we found is a nice co-localization between BGLUT2 and VGAT, okay? Showing that, I mean, we can really show that the axon buttons, they express both vesicular transporters, okay? This is one kind of analysis. Of course, to label the entopeduncular nucleus terminal, he injected a virus expressing a reporter under CRE, blah, blah, blah. The same I mentioned before. Sorry, my Spanish is filtering. Same I mentioned before. Then in a more recent work, also with Frank, but now in his own team, what he did is to label, for instance, lateral hypothalamic neurons. So then we analyze these terminals in the VTA, terminals of these neurons in the VTA, and we analyze input specific from these neurons to dopamine neurons in the VTA, and how these are changed after stress, in particular food stress exposure. And in this case, we didn't analyze we were not focused on the presynaptic inputs or the amount of inputs, but also we were interested mostly in the postsynaptic aspect of those inputs. And in particular, we were analyzing expression of GLUA1 of AMPA subunit, AMPA receptor subunit, GLUA1, and then analyzing the level of activation of those synapses by studying the presence of this subunit that we know are necessary when you have an active synapse. Okay, so to conclude, and then to leave some space for questions, if you have any question, AT is a very sensitive and reliable quantitative approach to study input specifics and active circuits. It's highly compatible with stereotaxically inject viral tracers. For instance, I show you data with the YFP, tomato, and cherry, but I keep trying others like Venus. So this is important also to share. This is why we try to share this knowledge in our website, in arraytomography.org. The idea is to share this. If you try and it's working, let us know. If it's not working, let us know, and then we save some time. And then the other thing is the antibodies. I saw Christina talk way ago, but about this. So antibodies are very important for array tomography. If you don't get a nice immunolabeling, it's not going to work nicely. But then there is maybe some space of some testing on antigen retrieval or potential antibody enhancement with different techniques, maybe with some different modified fluorophores, etc. This is all kind of work in progress. Community is very, very small, so we are trying to expand this community and then to, let's say, to speed up on this potential things to do. And then, of course, thank you again for letting me to be able to participate on this. Very interesting. I'm looking forward for if you have any question, or please, I think if you Google my name, my email is there, so feel free to email me if you have any very, let's say, very specific and long question, just email me. But I'm happy to take questions.
Thank you, Mariano. Any questions? I don't. Okay, I have a question about the LR whites, that the flat embedding. So you did mention that you do only dehydrate to 70% alcohol, but you still bake it, right? So you think the effect on the fluorescence, the heat does not affect the fluorescence as much as the alcohol dehydration?
Yeah. At least I don't see my image. Okay. At least in my experience, yes. I go up to, let's say, I typically use 55 or 57 degrees for cooking, and fluorescence is there, very strong. Of course, some fluorophores are my favorite, or I would say stronger than others. But so far has been okay. Yeah.
Okay. All right.
But though alcohol, I have tried, and it really kills even the strongest YFP, mice. Is killing that. So that's it. Yeah.
Wow. Okay. Good to know. Okay. Paolo, you want to ask a question? Go ahead.
Yes, we were wondering how you elude the antibodies to do multiple rounds of labeling.
Yeah. Thank you for the question. It's a very good question. The elution is based in a solution that has some twin and some sodium hydroxide. But it's, I mean, I can give you a recipe, but it's really easy. It's like half an hour, 20 minutes. It depends on the antigen, because some antigens, of course, if you have tubulin or synapsin, all the tissue is full of it. So to remove everything takes longer, but some antigens is very quick and easy to remove. Then you have to go through again to prepare the sample again to be stable, to go for a second round and third round. But it's up to 5 rounds I did, and to put all together, and it's okay. Yeah.
Amalia, you want to go ahead?
Hi, Amalia Passoli from Rockefeller University, originally from Universidad Nacional de Córdoba, Argentina.
Hello, hola.
Nice to meet an electron microscopy from Argentina. I was also going to ask about I have never done array tomography. When you do illusion and try multiple antibodies, that this affect the section somehow, that does it damage the antigens that you should, for example, I will try first for the antibodies that are more scarce and for the strong antigens at the end, or
yeah, it's a very good
the
question.
same for all of them?
Yeah. No, usually, let's say I would say most of the antibodies I use, I kind of I've been historically moving them, back and forth, first round, second round, third round, and most of them are very strong, very good. It's true that if you have an antibody that is a little weak, let's say I wouldn't leave it for the last round. That is true. Even though I would say we did that, and actually we analyzed that, but with PSD95 labeling, there is like a canyon. But and in that case, antigenicity between, I think we did 2 or 3 rounds, it was like almost 99%. So, I mean, this meaning that the same pixels were labeled across different rounds, So very, very strong. But I agree that, let's say, if you follow a receptor or something a little bit, even for regular immuno, it's a little weaker, receptors or more like single molecules, no clusters of molecules. You have to be careful of that. Yeah.
Okay. Thank you, Maria.
Yeah. But the tissue is very nicely preserved because, let's say, if you do a contrast, the morphology looks very good across different rounds.
Okay.
Yeah.
Thank you.